students, we will continue our topic of biological oxidation, competition number 6.6. Today's topic is mechanism of oxidative phosphorylation, in which we are studying mainly the chemiosmotic theory. Transfer of electrons from reduced coenzymes present in the electron transport chain through to the oxygen is known as biological oxidation, and during that process energy is released, and that energy is trapped in the form of ATP by the process called as phosphorylation. So coupling of biological oxidation and phosphorylation is called as oxidative phosphorylation. Many theories have put forward to explain the mechanism of oxidative phosphorylation, but none of them explain it completely. But the chemiosmotic theory put forward by Peter Michel in 1961 was widely accepted, and we are going to study this theory in details. Chemiosmotic theory. It says that when electrons are transferred into the electron transport chain at various sites, energy is released in the small amount, and that energy is used to pump the electron from matrix to intermembranous space across the inner mitochondrial membrane against the concentration gradient. Due to this pumping, a proton gradient is created across the inner mitochondrial membrane. Now, if you see here, in the intermembranous space, the concentration of proton is more, and on the matrix side, the concentration of proton is less. So, proton tries to enter onto the matrix side, but inner mitochondrial membrane is impermeable to the protons. Hence, proton has to enter into the matrix through a channel called as ATP synthesis complex, or also called as complex 5. Now, this ATP synthesis complex is made up of two subunits, F1 and FO subunit. FO subunit is embedded in the inner mitochondrial membrane, whereas F1 subunit is present in the matrix, and a channel is present between these two subunits for the transfer of electron. When 3 protons passes through this channel of FO and F1, it get rotate and one ATP is synthesized. When proton enters into the channels of FO and F1 and passes through it, then ATP synthesis complex, there is a conformational change in the structure of F1 subunit leading to ATP synthesis. So this is the diagrammatic representation of F1 subunit. First, it is present in L form, less affinity. L indicates less affinity. So here ADP and inorganic phosphate binds. When the proton enters into the F1 subunit, suddenly L form is converted into T form. It is also called as high affinity form. In this case, the ADP and inorganic phosphate, which are away from each other, come close to each other, and that phosphate is incorporated or binds with ADP, and ATP is synthesized. When ATP is synthesized, suddenly this T form is converted into O form, and it releases its ATP. When ATP is released from O form, this O form again get converted into L form, and a new ADP and new inorganic phosphate comes and joins there and attach to the L form. When 3 electron enters again into the F1 subunit, again the L form is converted to T form and ATP is synthesized. So in this way, this process is going on. To convert the L form into T form, the 3 protons should pass through the F1 subunit. Otherwise, this L form is not converted into T form, and ADP is not converted into ATP. So the passage of 3 protons are very essential for synthesis of ATP, and these protons passes from intermembranous space to the matrix, because in the intermembranous space the concentration of proton is more, and on the matrix side the concentration of proton is less. Thus, when electron passes through the complex 1, 10 protons are pumped and 3 ATPs should be synthesized. 3 3s are 9. For 3 protons, 1 ATP is synthesized. When electrons passes through the complex 2, 6 protons are pumped out and 2 ATPs should be synthesized, right? So 1 NADH should give 3 ATPs and 1 FADH gives 2 ATPs. But this is not seen because leaking of electrons occurs through the inner mitochondrial membrane from intermembranous space to the matrix. What I have told you, that inner mitochondrial membrane is impermeable for protons, and hence protons will not travel through the inner mitochondrial membrane and they does not enter into the matrix. But this inner mitochondrial membrane is not 100% impermeable for protons. Small amount of protons passes directly from inner mitochondrial membrane and enter into the matrix, and during this process they bypass the FO-F1 complex, and hence ATP is not synthesized. It is considered that 1 NADH gives only 2.5 ATPs and 1 FADH2 gives only 1.5 ATPs, simply because some of the electron bypass the FO-F1 complex and they directly travels through the inner mitochondrial membrane and enter into the matrix. This is about oxidative phosphorylation. Thank you very much.